Good evening. Welcome to this evening's C2ST event. My name is Jay Walsh. I'm Vice President for Research at Northwestern University. I'm also one of the Board of Directors members for C2ST. C2ST was started about 3 years ago with the goal of bringing science and technology to the forefront of the mind, the consciousness of the public in the Chicagoland area. It's comprised of most of, what we think of all of, the major science and technology institutes and organizations in the Chicago area. As a board, we get together a couple times a year, a bunch of subcommittees that get together far more often than that, to put together the various programming that's involved with this organization. And it allows us to meet with each other and invite the public in, as we're doing tonight, to listen to some of the best minds in the area of science and technology that exist in Chicago, the world, the United States, and the world. We're also a member organization, so as you came in, you may have noticed, you can join. Joining allows you a few benefits, not the least of which is to get email on a regular basis and see the programming that we put together. One of the next programs that we'll have is When Crocs Ate Dinosaurs, which is, I think, this Sunday. This event is going to be held over at Northwestern University, here on the Chicago campus of Northwestern. And then one of the next ones after that is a completely different area. It's in physics. It's programming that we often do, type of program we often do, which is we join with another institution. This one happens to be the physics department up at the Chicago campus of Northwestern. The Howborn Symposium brings in world-class speakers. 2 years ago, we brought in Steve Chu, who's now Secretary of Energy. He's also a Nobel laureate and a few other things that he might have done in his life. But that's the level of folk that we bring in for this sort of symposium, and we welcome the public, as we did with Steve Chu. We welcome the public, and we partner with C2ST. So tonight, it's my great pleasure to introduce two really phenomenal scientists. First is Sam Stupp. We'll hear him, and then after Sam talks, we'll hear Jack Kesler. Both of these folks are impressively world-class. I know them as faculty members in my home institution, and sometimes when you have faculty members in your home institution, we have a saying in academia which is internationally renowned and locally unknown, and sometimes that really applies. Now, in my job, I'm not supposed to say that, and they're going to go and tell all their faculty that their vice president thinks they're locally unknown, but that is absolutely not true. But you know, what we often do is we often don't recognize the greatness that is right down the hall, and these two gentlemen are really just doing phenomenal work. When I travel around the country, I have questions about what they're up to on a regular basis. So let me introduce first Professor Sam Stupp. He has a bachelor's degree in chemistry from UCLA and a PhD in material science and engineering from Northwestern. Did that in the 70s. Stuck around and was a Northwestern faculty member in the material science department for a couple of years, and then went to the U of I down at Urbana-Champaign for almost a decade and a half. Where he rose through the ranks and was in the material science department down there, the chemistry department, the bioengineering department, and worked at the Beckman Institute. We lured him back to the Chicago area, back to his home at Northwestern, about 12 years ago now, 10 years ago now, where he's a Board of Trustees professor of material science. He's also a faculty member in chemistry, and also a faculty member in the medicine department. So not only is he in 3 departments, he's in 3 different schools, which means he reports to 3 different deans. And on top of that, he has a center which he runs, which is called the Institute for nanotechnology in medicine, and that reports to the Vice President for Research, that's me. So he has somewhere between 4 bosses and none, which is what you do when you rise up to these ranks. Your real boss is science, which governs what is really what you're doing, is you're driven by data. He's a member of many organizations, a fellow of the American Academy of Arts and Sciences, the American Physical Society, the American Association for the Advancement of Science, World Technology Network, and the list goes on. Many awards, which I'm not going to go through right now, and I'm going to stop talking so we can hear from Professor Sanderson.
Thank you, Jay. When I first came and saw him, he's one of my bosses, I asked him about my budget. So it's already been very useful this evening. Okay, so I'm here to tell you about nanotechnology and about the event when nanotechnology meets regenerative medicine. So let's start first defining things a bit. Nanotechnology, what is it? It's difficult to define, but it is a technology of very, very small things. And so what it means is that it deals with objects or structures that are 1 to 100 nanometers in dimension. And so what is a nanometer? Well, a human hair is a very small fraction of a millimeter in diameter. You can just do the experiment. I won't do it because it will hurt a lot on my head. But so that's a human hair. So a nano hair would then be something you create by producing 100,000 split ends on that single strand of hair. So that is roughly the scale of a nanometer. So this is why this is very, very small. And so, you know, why nanotechnology? Okay, so there is some history here about the development of nanotechnology in this country and in the world at large. But there are many things that led to the development of nanotechnology. One of them was a very interesting discovery that the properties, or a series of discoveries, telling us that the properties of matter can change with size. So if it's large is one property, if it's small is another property. And in fact, right here at the bottom of the slide, I illustrate this point. This is a nanostructure, a few nanometers in size, called a quantum dot. And here you see 4 bottles that have quantum dots suspended in some fluid. And you see that they look, they are different colors. And they are being illuminated by ultraviolet light. And the color that you're seeing is luminescence, okay? So they are emitting photons, they're luminescent materials, but this one is blue, this one is green, this one is yellow, and this one is red. And basically the nanostructures, the quantum dots, are increasing in size as you go from the blue to the red. So this is a very vivid picture of why the size matters so much. So in the nanoscale, you change dimensions, you change fundamentally optical properties. So that was a very important discovery that launched the field. But also in the last 2 decades or so, we have come to understand very well that cells, life, are basically highly orchestrated arrays of nanostructures, and the way they interact lead to life itself. So that means that putting together nanostructures in principle should help us build new materials, new devices. Perhaps some of them might be very small or might be very powerful. They may have new functions. And scientists around the world are thinking about the use of nanotechnology in many areas. Energy, medicine, personal safety, solutions to environmental problems. So there are many opportunities there. And of course, the other important point about why nanotechnology is basically this really is going to help us understand nature much better than we do right now. And so what is it that scientists do in this space, in the nanotechnology space? So scientists in this field, what they do is they design and they make nanostructures. And because of what I just told you, controlling the size and controlling the shape even is important. So you can get different properties that way. And you can also then encode functions in these nanostructures. So you think about what special properties could nanostructures have if I design them to have a specific composition and a specific architecture. Another very popular activity in this field is to actually probe the nanostructures. And the new thing, the cutting edge as we speak right now, is not so much to make a nanostructure and design it and have it and make something of value because of the functions. The cutting edge is learning to organize them into larger things. So things that are now microns in size or things that are macroscopic. I'll give you an example of that. And the bottom line here is that there is a lot of science behind all of these tasks and lots of potential technology as well. So it's a very exciting direction. And so what are those functions that we are looking to develop? Well, there are many, but I just want to give you an example. A nanostructure can be a sensor. It could be a very special sensor or something you're interested in knowing. And this is a capability that maybe we did not have before. A nanostructure might be a drug carrier that knows where to go. This is one of the hottest concepts in the field right now, developing vehicles for drugs that when we swallow them or when they are injected into human bodies, they know exactly where to go, which organ to go to in order to have a therapeutic effect. And also, they could be electrical wires, and this would help us build special devices. And in red right here, because this is the field I want to discuss with you, they could be maybe activators of cells. And this is the idea that we had 10 years ago, that we wanted to develop nanostructures that could talk to cells, that could signal cells. And so the target, and this, all of this work started when I arrived at Northwestern in 1999. My hope was to launch a program where we would go after regenerative medicine as our target. So we had, in the 90s, developed some of the first examples of nanostructures, especially of the variety that has organic composition. And so we wanted to think about nanostructures that would help regenerative medicine. And of course, why should we care about regenerative medicine? Well, it's pretty obvious. This list is an interesting one. These are all areas that we are interested in my laboratory at Northwestern. And these areas are all at different stages of development. You're going to hear tonight, after my talk, you're going to hear about our work on spinal cord injury, which we have done jointly with Jack Kessler in the Neurology department. But there are other targets, for example, heart regeneration, or even strategies for Parkinson's and Alzheimer's disease, the possibility of addressing diabetes problems, and very importantly, also the development of the regeneration of cartilage, because after you turn 18, there is no way that you're going to regrow your cartilage. And if you don't have good, healthy cartilage in your joints, you are going to be a very unhappy individual. And many of these lesions proceed on to osteoarthritis. So it's very important to understand how that regeneration takes place. We are very interested in bone, and we are also interested in dental tissues, for example, regenerating enamels that can help people die with their own teeth, which nobody does right now. So these are all important targets in regenerative medicine, and we have tried to study this problem, that is the idea of activating tissues to regenerate using nanostructures. The idea was the following. So this is a Picasso representation of of the environment outside a cell. So the cell has a membrane, and that is down here. If you start exploring the so-called extracellular space, you find that there are lots of fibers in there. It's full of fibers. There are fibers of many different kinds. There are proteins. These color objects here are all proteins, and they are bound to these fibers that exist naturally. For example, this red fiber that you see here. So our idea was to somehow invent molecules that would self-assemble into nanofibers these silvery things here. They would invade the space, self-assemble from soluble molecules, soluble in water, and that they could then self-assemble into nanofibers. And those nanofibers, 6 to 8 nanometers in diameter, would emulate collagen fibrils, which are natural components of the extracellular matrix. And then we would, by design, put signals on those fibers that would help start processes important in regeneration. By definition, in regenerative medicine, you're trying to do something which does not occur spontaneously. That's, it's by definition, you're trying to jumpstart something that doesn't occur if you don't do anything about it. And so the molecules that we developed, and now it's been roughly about 8 years since we first reported them, are very special molecules which have this structure that you see on the slide. They are basically soaps, okay? So you put them in water, you'll see the bubbles, just like as you put dial soap in water, you'll see bubbles. They are soap molecules, they are surfactant molecules. But they're very special structures, and our groundbreaking contribution here was to develop specific peptide structures. This is a peptide segment, this is a very hydrophobic tail, which makes it a real good soap. We developed the structures that could actually lead to nanofibers by self-assembly when we inject these molecules into physiological environments. So they self-assemble into this nanofiber, which measures about 6 to 8 nanometers in diameter, but it can be extremely long. It could be microns in length. And then when this self-assembly occurs, the biological signals, which we put them by design right here at the end of the molecule. So when this self-assembly takes place, the surface of the fiber has now all of the signals that we need for regenerative processes. And so we understand now very well the details of how this actually occurs. And so, for example, here, this is now a real simulation. This is not just a cartoon, but it's a real calculation using something called Monte Carlo and stochastic dynamics. And we understand exactly how these soap molecules get to be these cylindrically shaped nanostructures. And in very recent work, and this is, you see here, and I wish we could maybe lower the lights a little bit so you could see this better. This is a fantastic electron micrograph of the cryo type, which means, yeah, that's much better. So this means that we basically freeze the solution after self-assembly has occurred, and we get to see, with high fidelity, we get to see what these nanostructures really look like. And as you see here, they are extremely uniform in diameter. And they bundle up sometimes. And if we had here micrographs of natural extracellular matrices, the fibers of the extracellular space, they will look very similar to this. But of course, these are completely artificial, and they're loaded with signals by design. We can customize them for the various targets that we have. And also, they're biodegradable, which means that after they jumpstart some regenerative event, they can disappear within a few weeks. They can be basically be biodegraded into nutrients, and they don't leave any traces. So it's very different from putting carbon nanotubes or quantum dots or any other of the nanostructures that you might have heard about. Those don't degrade. These do degrade and disintegrate into nutrients. And so they form very interesting structures. And also, one of their characteristics is because they form these networks that you just saw on the previous slide, then when those networks form, they actually tend to gel the water in which the molecules are dissolved. So when I turn on the self-assembly, and I do that by introducing electrolytes, by doing certain magic procedures, then the self-assembly front occurs, so a network is organized, and that turns the liquid into a gel. If I then reverse the process, I can get the gel to go back to being a liquid. And one of the interesting things about these nanostructures is that when they form into these networks by adding physiological things like, for example, cell culture media, they form the networks right, in very, very short periods of time. So that means that if I were to inject them into a human being, in principle, you could expect that they will grow in situ, surrounding cells, they'll make networks, and they will get trapped at the site of injection. And this is a characteristic of their shape, because they are very, very long and very thin, so they trap each other into a complex network, and that's why they are localized at the site of injection. Now, instead of the Picasso, you see a bone cell. Here, we zoom in, and you see the membrane of the bone cell, and where we've treated the cell with a solution of the molecules, and you see the fibers right here in the cellular space hugging the cell. And so if those tiny fibers have signals that can talk to cells, they're ready to get, in this case, the bone cell to do something. Okay? So the platform, this platform of nanotechnology for regenerative medicine is pretty broad, and so this is a gallery of about 10 molecules. There are long ones and short ones, and even some with branch structures. And so we basically developed the code. We invented the structures that basically tolerate many different compositions. Literally, this could be thousands or tens of thousands of compositions. And when you treat them by adding electrolytes or injecting them in physiological environments, they will always self-assemble into these nanofibers. This particular one has two molecules, has a very long one and then a short one. And the long one sticks out, making this hairy nanostructure. Well, it turns out this piece that sticks out happens to like certain proteins that make bone regenerate. And so basically you customize them to the application that's needed. So I want to illustrate, without giving you all of the details that that are behind this, but in cartilage repair. So if you have a tiny lesion in your knee like that, this is a multi-billion-dollar business in this country, and nothing really works. They tell you it works, but it doesn't really work. But you have lesions, and so what they do is they remove the lesion here, they remove the cartilage, and then they do something which might sound a little primitive at first, which is they drill holes into the underlying bone, right? So in the joint, you have a bone covered by cartilage, which is key to the function of the joint, and the surgeon drills holes until the bone bleeds. And supposedly the bleeding, the hypothesis is that the bleeding exudes all these proteins and stem cells of various kinds, like a group called mesenchymal stem cells, which can turn into presumably cartilage. Well, as it turns out, when they do turn into cartilage, they turn into the wrong kind of cartilage, called fibrocartilage, and so the function of the joint is definitely not achieved. So that procedure, as it stands right now, doesn't work. So we have recently shown that if we do the same procedure, not in humans, but these are in animal models, but if we do the exact same procedure that orthopedic surgeons do, then, and then we put our nanofibers on top of the holes, then you get the right kind of cartilage, which is called hyaline cartilage. It has the proper morphology, and this slide proves that. So you see here, in these two cases, in this area that you see here, that's where we would have drilled holes. So now, 12 weeks later, cartilage has grown here, and this one is okay, but it's not great. This one is fantastic by all counts. So this whitish tissue that you see here is authentic cartilage, as demonstrated by this histological slide. So to get perfect cartilage, you need to see this red stain, which is stained for the glycans, and the cells of the cartilage have to be aligned in rows like this, not like that, but this way. So this blue area in the defect is more like fibrocartilage. This is collagen. This is the good stuff. And the good stuff is is regenerated by this nanostructure, which in this case has a tether here that likes a certain protein that is important in the regeneration of cartilage. For example, in turning mesenchymal stem cells into chondrocytes, which are the cells of cartilage. So this is a very good recent example. And then we have others. For example, in this case, we've shown that we can develop another nanostructure, different chemistry. So every time we're doing something different, we're changing the chemistry, but we're keeping the architecture of the nanostructure, the cylinder, the nanofiber. And here, for example, we have a specific type of signal that makes bone marrow-derived cells proliferate like crazy when surrounded by our nanofibers with specific signals. And so this is going to be very useful, for example, in certain models where they inject bone marrow-derived cells into the heart to attempt to rescue myocardium after infarct, and in chronic wound healing for diabetic patients and cancer patients and many other situations. So this is a fiber customized for regeneration in those targets. So I want to finish up to showing you what I said at the beginning, that the cutting edge in nanotechnology now would be learning how to organize things into larger arrays. This is a very recent example, and I would just show you that if you take a solution of those magic molecules I've been talking about, but then you bring in a polymer in water, and these two droplets touch, in milliseconds you pull out a bag, basically, which encapsulates the polymer, which is fluorescently labeled in green, so you can see it. And then when you start exploring the structure of this membrane, what you find is that when the two liquids touch, these nanostructures form, so lots of them, all going in the same direction. But then the polymer, which is in the other liquid, begins to pull through little tiny holes and nucleates more nanostructures. So now you have trillions of nanostructures being formed and all vertically aligned in a very specific way. You might be wondering, well, why do we care about this? Well, it's very important because this membrane is actually very biomimetic of natural tissues, and they're composed of arrays of nanostructures. I told you that cells, for example, are highly orchestrated arrays of nanostructures. And so if you then take a look, I showed you the cartoon, but then if you take a look at the electron micrograph, you see the formation of this membrane with all these nanostructures vertically aligned, perpendicular to the plane of this membrane that made that sac. So here's the idea. The idea is, can we use this process to make something that looks like a cell, make an artificial cell, right? So we have already started doing these things at Northwestern. This is now a collage here of real cells and artificial cells. So for example, this guy right here, or these guys over there, these are artificial cells made by this process, which organizes nanostructures into a membrane. And so with this, we hope to generate many new concepts that we can use for therapies and even for concepts outside of medicine to deliver to materials important molecules, and using the biomimetic sort of biology mimicking approach of doing things with cells. Okay, so the other one, which is a very recent one, is we have also learned to make large arrays of the nanofibers, and you see one in a cartoon here. And this array is actually really organized. So each one of these dots, each one of these red dots is one of our filaments, one of our fibers. And they are organized into a crystal, actually. This is an actual crystal. And for example, there are places in biology, for example, the cornea in your eye has collagen fibrils. in arrays that are organized pretty much like this. So we're very excited because we are finding ways to organize nanostructures in ways that can produce materials, new materials that look like natural tissues, and this will lead to many new ideas. Okay, so as we get ready to listen to my colleague, Jack Kesler, with whom I've had probably the most exciting collaboration I've ever had in my scientific career, I want to show you, just to introduce what he's going to be talking to you about, that there is one fiber with the many compositions that we've made. One of them has a signal which is called IKVAV. Those are the letters of the corresponding amino acids. This signal is very important for everything that happens in the central nervous system. Very important during brain development. It is important in the growth of neurons. It's important for repair of the spinal cord. And so these fibers have been quite exciting to work with. We had made initially a discovery that they guide the differentiation of neural stem cells. This was the beginning of our collaboration back in 2003 or 2002. And we discovered that, in fact, these fibers that you see here, these are the neurites of neurons that are formed as a result of the differentiation of a neural stem cell into a neuron. And this process is mediated by the fibers, by the functional fibers. So before I turn the podium over to Jack for his part of the story, focusing on spinal cord injury and stem cells, I just want to say that everything that I've told you about is not done by me, but it's done by my students and my postdocs. And so, so this is one recent picture, and they're, of course, the ones that do everything. So I just want you to recognize that all the wonderful things that you hear about in science, in scientific research, is actually done by young people. People that are between 20 and 35, basically. And of course, without them, none of this would ever happen. So I think with that, I will thank you for your attention and hope that you will enjoy the second chapter of the story by Jack Kesler. Thank you.
Well, we switch over laptops here, or not. I'm going to let them do the technology, even though I'm the engineer, and introduce Jack Kesler. There's a variety of ways of doing this introduction. I can say that Jack is the Davee Professor of Stem Cell Biology, the chair of the Davee Department of Neurology. He's also the director of Northwestern Stem Cell Institute, part of our Neuroscience Institute, and clearly collaborates with Dr. Stupp. You know, I sort of made light of the fact that Dr. Stupp had appointments in various departments. I want to circle back to that, kind of do this on purpose, circle back and say that some of the most interesting work that's done in science, in medicine, and frankly in most other fields is done at the interfaces between traditional fields. So the fact that we have a faculty member here who's in 3 different fields and doing some fantastic work and collaborating with somebody who's in a completely different field, in neurology in this case, is not by accident. This is actually done by design at most universities now. And you will probably notice this if you read in the scientific literature and you look at the people who are doing this work, it's at the interfaces. And I bet if you look at these students, this group of 30-ish people that are in your lab, they're from they're from those 3 departments and more. And that collaboration of these students across these departments is absolutely vital. So getting back to Jack Kesler, I could also talk about unknown lands. Jack had a movie that was made of his life a couple of years ago. Tara Incognita, which is a movie that has a part of its title is The Promise and Peril of Stem Cell Research. Jack got into the area for a variety of reasons, and in the movie, if you wish to go find it, a good movie to go rent on a weekend, got into this because of an accident that his daughter had at the age of 15, a skiing accident that left her paralyzed from the waist down. She has since gone on and done pretty well. She went to Harvard, where she was the coxswain of a heavyweight crew, and many of you know about crew, that's a big deal. She then went on to the London School of Economics and is now back at Northwestern, where she is a medical student. But that paralysis, like many people in life, if you sort of knocked upside the head by some major event, you refocus. And Jack is refocused on using stem cells, as we will see, to cure the condition of, or in many neurologic conditions, but certainly the one that his daughter is facing now, which is paralysis. I'm not going to talk any more deeply about this. I don't want to either steal his thunder or ruin the story, because that's really what would happen. And I'll turn the microphone now over to Dr. Jack.
First, I have to apologize a little bit for sounding like a frog, but neither nanotechnology nor stem cell biology cures the common cold. So here I am. I'm going to talk to you, Dave, about spinal cord injury. So it's important first to understand about the spinal cord, what exactly it is and what the problem is. So the spinal cord is the part of the nervous system that basically connects the brain to the rest of the body. It sends all the motor signals down to nerves that allow us to move. It carries all the signals back up from our limbs to our brain so we can sense things. In essence, it's a gigantic cable. And the problem in spinal cord injury is that the cable has been cut and the fibers are trying to regrow. So you can see, actually on this picture, in the dark there's an interrupted group of fibers that are trying to regrow. In this particular case, the head would be down there, the legs would be down there, and this is the spinal cord running this direction. Now those fibers actually have the capacity to regrow, but they don't after the spinal cord is injured. It simply doesn't happen. Why? Number one, there aren't really the right signals to tell them to grow. Two, there are actually signals that keep them from growing. And number three, there's a problem of what's called the glial scar. So if you look at the blue stuff there, that's sitting there with the middle of all the cysts, that's a scar that forms that acts as a physical blockade, preventing these fibers from growing. So it's a very complicated problem. These are the kinds of things that we have to overcome. Now that glial scar is made up of cells that are called astrocytes. So as I mentioned to you, they act as a physical barrier to outgrowth. They also mentioned there's some inhibitory molecules, they're chondroitin, sulfate, proteoglycans, the name doesn't matter, but it actually traps some of those molecules and keep the fibers from growing. So you would think then, oh, it's easy then, just stop all the astrocytes. But one of the problems is that they also do good things. They limit, when the spinal cord is injured, bunches of cells rush into the nervous system from outside of it, inflammatory cells. There's something called a blood-brain barrier, which normally protects the nervous system, and that's ruptured. So what we really have to do is think about how can we keep the good parts of that response and get rid of the bad parts. So if we look at a spinal cord that's acutely injured and then chronically injured, I think you can see they look very different. On the left is an acutely injured one. If you look at the middle panel, the astrocytes, the cells that form that glial scar, are in red. And I think you can see that they're spread out over a wide area around the injury site. If you look at a chronic spinal cord, I think you can see they've narrowed in now and they're a narrow little band. Now on the bottom, that's a marker for inflammatory cells, which are a bad thing to have in the nervous system. You can see acutely all these cells have rushed into the spinal cord, which is a bad thing. And so the spinal cord, to try and heal itself, walls them off with these astrocytes. And so if you actually look at the cells, is there a difference between these cells in the acute and the chronically injured spinal cord? There's a vast difference. If you look on the acute side, that will be on the left there in the middle panel, the cells are bigger than a normal cell compared to the top. If you look at the chronically injured one, there are many more of them. So what that means is in the short term, it's okay to let them get bigger, but we have to stop more of them from being generated. Okay, so the key to blocking the glial scar is preventing more of these cells from being generated. So now I'm going to show you just to repeat a few of the slides you just saw and try to put them in the context of repairing a damaged spinal cord. So how are we going to think about changing the environment inside the spinal cord so those fibers will grow? Well, this is where my collaboration with Sam began. I have to tell you, it's one of the most exciting collaborations I've had also. It's just a joy to be able to work together with him. And he told you about one of these molecules that he made that has the sequence IKVAV. And he told you at the time that one of the things it does is it makes neurons put out processes, right? So that's the green that you can see on the left-hand side. So that's a good thing. The fiber tracks we want to get to regenerate are neurons, and we want it to put out the processes. What it also does is you'll notice on the right-hand side where you don't have the gel, you see some red cells. Those cells are the astrocytes. Those are the cells that form the glial scar. So in addition to making fibers grow, this material prevents those astrocytes from forming. So he showed you how these materials self-assemble, and he showed you how they can form a whole network of fibers like this. So now think about the injured spinal cord before this scar forms, injecting this material into the spinal cord. We seem to want to connect. Injecting this material into the spinal cord, fill any cysts that may be there, and prevent these astrocytes from making this scar. So, to be able to does this kind of material work in spinal cord injury, you need a model of spinal cord injury. So the model that we used in our first studies was with my colleague and collaborator, Michael Failings from Canada, who developed a very carefully calibrated clip compression model. So what you're seeing is a spinal cord shown on the left-hand panel on the bottom. And on the right, you're showing the spinal cord after this clip is put on, closes over the spinal cord with a very, very carefully calibrated pressure, and you keep it on for a known period of time. This is the kind of injury that happens to human beings. In human beings, the spinal cord is not typically cut, it's compressed, just like this. So this is a very good model for human spinal cord injury. Then what we wanted to do was intervene with a time course that would be feasible for human beings. So it's quite feasible to think that a patient could get to a hospital, be prepared for surgery, and 24 hours later, we could think about treating them. So we initiated the treatment. We injured the animals, allowed them to recover, waited 24 hours, and then went back, and in the data I'm going to show you, treated them with the material that Professor Stupp described to you. So the first question, does it prevent that scar from forming? Well, you've already learned to look at this red staining for the molecule called GFAP. That's a measure of the scar. On the left-hand side is an animal that's not been injured. In the middle is an animal with spinal cord injury that wasn't treated. And on the right-hand side is, my problems. On the right-hand side is a animal that was treated with a gel. And I think you can see that the cells are bigger on the right-hand side compared to the control, but there really aren't more of them. If you look at the one in the middle, you can see that there are many, many more cells. That is, what we've been able to do is inhibit this scar from forming. Those of you who like to see things in a graphical form instead of just looking at one picture, this is looking at a whole series of experiments. And I think you can see we haven't completely blocked the scar, but we've largely blocked that scar from forming. Well, now we get to the more important issue. Are we really getting regeneration of fibers? Now, how are we ever going to really know that fibers have regenerated? The way we do it is what's called track tracing. 3 months later, we go back to the same animals, we go back up into the brain, and we inject a dye into the brain so the neurons that are in the brain can pick that dye up, and it will be carried down those fibers in the spinal cord. So we can actually look at individual fibers to see if they're growing. In the same way, we can inject a dye in the lower part of the spine for the sensory fibers to look to see if fibers are growing up the spinal cord. Then what we have to do is go in, section the spinal cord, and my graduate students have to do a reconstruction of the spinal cord. This picture took my graduate student 3 months to do, to give you an idea of exactly how long it takes to do the reconstructions. So this is a reconstruction. What you can see on the far left is a reconstruction. The colored lines are fibers in an untreated injured spinal cord. And I think you can see they're growing up to the dotted line. That's the area of the lesion, and they stop. That's what everybody finds. In the second one, B, is a tracing of those fibers in an animal that was treated. And I think you can see they're not only growing into the lesion, they're actually growing through the lesion and outside the other side of the lesion. To the best of my knowledge, this is the first time anybody's ever been able to show this with motor fibers. So now one question that comes up that's actually a very important question. Did we actually make fibers regenerate, or by going in and treating, did we just perhaps prevent them from dying in the first place? What's the possibility? So to answer that question, we went back in 2 weeks in some animals before they would have had a chance to regenerate, but after they would have died. And that's shown graphically at the bottom. There are just no fibers at all. Compared to the right, when we wait 11 weeks, we have about 40% of the fibers that grow through the lesion. We did the same thing looking at sensory fibers, and we found the same kind of thing. That is, the sensory fibers in an untreated animal grow right up to the lesion and then they stop. The sensory fibers in the treated animal go through the lesion site and out the other side. Interestingly, motor fibers are normally more difficult to get to grow. We actually had fewer sensory fibers. We had about 15% grow through. The good thing is estimated you need between 5 and 10% of your sensory fibers to have relatively normal sensory function. So there's a very good suspicion that this would be sufficient to restore the majority of sensory function. Now you have the other critical question. What about behavior? Does it really make a difference to the animal? Well, this is a scale. It's called the BBB scale. I put this up for those of you who don't like to look at animals, so you can close your eyes for the next 2 slides that I'm going to show you, because I'm going to show you a typical animal that was injured and not treated, and then a typical animal with an average score that was treated. So this is an animal that's treated. You can see there's a little spasm of the legs, dorsal posturing of his hind limbs, the legs, and I think you can see there's no coordinated movement at all. And we are very specifically modeling a severe spinal cord injury. This is an animal that's been treated. It's by no means perfect, but I think you can see there's now coordinated movement of the legs and some weight bearing. So we certainly have not cured spinal cord injury. I wouldn't want anybody thinking that we think that. We don't. But what we can say is that we can give quite a striking behavioral recovery. If it's not at the beginning and the end, it's at least the end of the beginning in having a rational treatment for spinal cord injury. So we've seen that we can reduce the glial scar. I didn't tell you about changing cell death. We can get motor and sensory fibers to grow through, and we get better function. Well, we really want to think about using this in human beings. So we have to say, well, is this just because it worked in this one kind of mouse? Does it work, or is it just the kind of model we used? So there's another way of doing spinal cord injury, which is actually the way that's done in most laboratories. It's called a weight drop model, where you actually lift a weight up, that's the pin, and then you drop the weight on the spinal cord, like that. So we set up the weight drop model, looked at a different species of the rat, that's shown in the bottom, got exactly the same result to the rat. We looked at a different species, different strain of mouse, got exactly the same thing. So in 2 different injury models and 2 different species, we saw the same thing. Now, it's important to begin to try to understand the mechanism of action. How's it actually doing this? If we really want to go in and hone this therapy down, we have to kind of understand that. And I hope this doesn't get too technically detailed, but I want to tell you a little bit. The first question we said to ourselves, well, you know, here it is. We have a material that's got this IKVAV sequence, and this sequence was chosen for a particular reason I'm going to tell you about in a second. But do we really need that sequence? Is that what it's all about? So to answer that question, Professor Stupp's lab made another gel that instead of being IKVAV was VVIAK. Okay? It just scrambled the sequence. And the graph on the right shows you on the blue bar is the amount of glial scar in a normal animal reduced with the IKVAV, but actually worse with the other gel. So it absolutely depends upon that sequence. Now, any molecule that interacts with a cell, or at least most of them, that exert an effect on the cell, work on something called receptors. That is, they're specialized structures on the outside of the cell that interact with the molecule and carry a signal to the inside of the cell. There's a whole set of receptors that are called beta-1 integrin receptors. And some data came out suggesting beta-1 integrin signaling is a signal that works on neural stem cells. So the IKVAV sequence was known under some circumstance to interact with beta-1 integrin. So we said, okay, if we go ahead and we take neural stem cells, remember you saw Professor Stupp showed you a slide of what it does to neural stem cells, showed it, we said, what does it do to beta-1 integrin? And on the left-hand side, beta-1 integrin is in red. On the left-hand side is a control one. In the middle is one with a gel, and you can see all that red. What this gel does is it increases this beta-1 integrin. So, then what's actually happening to those progenitor cells? The ones that are differentiating to neurons are putting out a lot of processes. Remember I showed you the green in that slide? But what about the rest of them? Well, it's a very complicated slide, but the important thing is to look at the second set of bars, GFAP. That's the marker for those astrocytes, the cells that form that scar. And the gel, shown in red, keeps the stem cells from doing that. We already know that. The bars on the right are markers of stem cells. That tells us what it's doing is keeping the stem cells as stem cells. Now, the spinal cord actually has a whole population of very specialized kinds of stem cells in them that are normally committed to becoming glia. Those are the cells that are generating the scar after spinal cord injury. That suggested to us that what was happening is the gel was actually acting on the stem cells native to the spinal cord and signaling through this and keeping them from becoming the glial scar. So we wanted to prove that. So the first thing we did is we said, well, okay, let's get rid of that receptor. And we did it through a complicated technology called a Crelox technology. You don't care about that. What happens when we get rid of that receptor? That's that red bar. They all become astrocytes. So that tells us, yeah, wow. In fact, this receptor is necessary to keep them from becoming astrocytes. The next question then is, okay, this receptor is necessary to keep them from becoming astrocytes, but does it actually control astrocytes and actually inhibit them? And so what we did is we made two forms. One form of this receptor that always signals, one form of this receptor that can't signal and gets in the way of any signaling. And we see exactly what we expect to see. That is, when we block the signaling, that's the one on the right, you get all these astrocytes. When you stimulate the signaling, you reduce the number of them. So, here we are at a convergence of two technologies. We have nanotechnology. We used it because of studies that began with stem cells that suggested to us that we might be able to regulate some of the important things in spinal cord injury. And what we have found is, it turns out, this nanotechnology, this material is working by acting on stem cells that are already present in the spinal cord, increasing this beta-1 integrin that I told you about, and signaling to them and keeping them from becoming astrocytes and keeping them from becoming the glial scar. So when everybody thinks about stem cells and utilizing stem cells and manipulating them, most people think about injecting them into the body, transplanting them. In point of fact, this is one example, and I could give you some other examples where we're actually stimulating them positively. The real holy grail for stem cell biology is to manipulate the stem cells that are already present in our nervous system and get them to do the things that we want to do and stop them from doing the things that we don't want them to do. So, a company's been formed, a company called Nanotech. Our hope and our goal is to be able to do the studies that are necessary to take this material to the FDA and get them to approve this material for a clinical trial in human beings. We're very excited about it. We don't for an instant believe that this is going to be the entire cure. Spinal cord injury is very complicated. It's going to require a number of things, but this is the first thing that's come along that has the real possibility, as far as we can see, of actually getting fibers to regenerate. As Professor Stupp did, I have to thank all the people involved in this, the members in my own lab, the materials science group headed that he heads, as well as a number of my other collaborators. The point being that science is always a team effort. I don't do any of this myself either. It's all the young people in my lab also. So thank you for listening.
So now we have time for questions, and I'll open it up to our audience. Great.
I guess this is primarily for...
There's a microphone in the middle so that everybody can hear it. Would be great.
Yeah, hi. I guess this is primarily a question for Professor Stupp. So you mentioned these sort of nanofibril structures, and that you also have receptor molecules that are linked to them, or some kind of molecule.
Sorry, we got distracted by the...
Okay.
Okay, could you repeat the question?
Sure, yeah. So you mentioned with these nanofibril structures that you have some kind of a promoter molecule that's linked to them. So I'm just wondering what effect is you're seeing is from the molecule itself, and what effect is from just having the sort of fibril structures there?
Yeah, that's an excellent question, actually. Let me stand over here. Yeah, so we know that the way we probed that question in the original work is we made the fibril, which had varying amounts of the signal, starting at very low levels and then gradually increasing the number of signals. There was a second molecule that had no signal. And then we found that this effect, the bioactivity effect that we've been describing, actually depends on the density of the signal. And it is most prominent at fairly high levels of the signal on the fiber, say around 50% or so. So that tells you that the density of signal is very important, and certainly the density of signal is something that is enabled very well by the supramolecular, by the fiber architecture. But at the same time, we also probed the signal itself, just the molecule, not the fiber, but just the molecule, and we did not find the effect. So that means that you need both you need the signal, you need the density of signal in the way that the fiber is presented, and you need the fiber, the nanofiber architecture.
I'll comment just briefly with respect to the biology that we specifically studied. I showed you one piece of data, actually, where we changed the sequence from an IKVAV to a scrambled sequence. It was absolutely dependent upon that sequence. We've used quite a number of other sequences, an EQS sequence and RGD sequence. I also showed you data indicating that I didn't show you all the data, the effects of the gel are absolutely dependent upon the presence of the receptor for IKVAV, that is the beta-1 integrin receptor. So I showed you a Krelox experiment. If you take those cells, eliminate the beta-1 integrin receptor, the gel no longer exerts its effects. So if you change the signal, it doesn't exert its effects. If you get rid of the receptor, it doesn't exert its effects. I think that's pretty good evidence that it's absolutely dependent upon the signal. Now, the other piece of the story. If you take the IKVAV peptide itself, you can dump huge quantities on cells and it does nothing. Absolutely nothing. So it is, the biological actions are dependent upon that signaling sequence, but it's also dependent upon the presentation, the geometry of the presentation of that sequence, and the peptide by itself will not exert the effects.
So you need the nanotechnology part.
So you need the structure to present it, but you need that sequence.
It is also, it doesn't work well either when you use the protein, the natural protein, that bears the signal, laminin.
would this have application to other therapies? For example, the nerves, I'm sure the nerve sheath, like in multiple sclerosis, could that repair that sheath?
Right, so the cell that's abnormal in multiple sclerosis is a cell called an oligodendroglial cell. And I didn't show you those data, but we have a variety of other manipulations. The cell that forms the glial scar, I mentioned to you, they're actually committed to becoming glia. They have two choices. They become astrocytes, the ones that form the scar. They can also become oligodendroglia, the cells that die in multiple sclerosis. So the answer is yes. In fact, in this particular material, it pushes those cells to become oligodendroglia rather than becoming astrocytes. I don't believe this particular material is going to be the way to treat multiple sclerosis, because that's a disseminated abnormality in the nervous system. But there are other signals that can push the endogenous stem cells that are present in the nervous system to become oligodendroglia, and quite a number of laboratories, including my own, are studying that process.
Question in the back.
I know this may not be your specific specialty, but maybe Dr. Stupp can comment. What about like other than nerve problems, like cardiac tissue or muscle tissue, or finding that receptor promoter kind of combination you guys sort of on?
Right. So I think every target in regenerative medicine requires a specific design. So I can illustrate, for example, in our efforts to rescue myocardium as one of the regenerative targets after infarct, the approach that we used is we developed a fiber that tends to jumpstart the growth of new blood vessels by the process known as angiogenesis. So the signals that we put on the surface of the fiber were signals that could activate the proteins that eventually lead to angiogenesis, that is, rapid growth of blood vessels. We have actually tried the idea in animal models, and we got improvement in ventricular contractility in an infarct model as a result of the fibers being present.
What about, did you face any issues with like rejection of these artificial polymers or what about that?
Yeah, we've never seen any evidence of immune rejection, and that doesn't mean that it cannot occur. There could be, because there's an infinite number of fibers that you can make. You can design, so many that there may be a fiber that is indeed immunogenic. But the ones that we have studied so far, we've never seen immune response. And the reasons why we don't see it may be many, but one of them is the idea that we are always targeting on the surface of the fiber. We target designs that bear natural signals, not foreign peptides, but natural signals. But even when we have used artificial peptides to bind proteins and so forth, we've not seen an immune response. This may have something to do also with the size of the peptides or even the architecture of the fiber.
I was actually curious about the cells that you engineered. I saw that there were some cancer cells that you had duplicated using this nanotechnology. Or there were, in any case, pictures of cancer cells, and there were pictures of blood cells.
Oh.
Was that, maybe I was reading too much into the diagram. Was
Yeah,
that,
I think, you were reading too much into the diagram.
I wasn't sure.
So what I tried to depict on that slide
Yeah.
is the notion that we can generate objects, micron scale objects that look like cells, and in fact, they also have the mechanical behavior of cells. They are similar mechanical properties as normal, as natural cells. And so I was just showing a whole variety of different cells, trying to match the architectures that we actually produced artificially.
But there's no, I don't know, I almost was
No.
like a vaccine or like
No.
what would you use? to
That we are too far from that. These are very primitive artificial cells, I would say. They're prebiotic artificial cells. Very, very primitive, but we are starting to put functions into them, and I think slowly the functions will become more and more sophisticated, and we'll get somewhere.
Is there a potential application here in dialysis that you could immobilize something on a fibril and then flow past it, biological fluid in a circulating system, and remove from the body.
You mean to remove
Factors that would
factors?
that were harmful, that were causing aberrant growth.
Yes, I think in principle something like that is possible, and of course in dialysis they use fairly inexpensive polymers in the membranes. They're cellulose derivatives. They're very inexpensive materials, and they do an adequate job. Of course, companies like Baxter are always trying to improve on dialysis membranes, but I think it's possible, but the question is there a real figure of merit here, right? You have to gauge the cost of what it would cost to create these membranes, and how much improvement you would get in the dialysis process. So, in principle, yes, you can design the fiber to absorb, to pick up specific molecules, specific factors.
Good question over here.
Has anything been done in the area of stroke? Because it seems like there hasn't been too much done in 30 years to repair the damage.
Actually, there's a lot going on in the area of stroke. I mentioned to you that the adult brain actually turns out, somewhat to our surprise in recent years, to have a very rich supply of stem cells. And those stem cells have the capacity to generate all the cells that are in the brain, they just don't. So there are quite a number of studies going on right now to find ways to mobilize that existing population of stem cells to get them to migrate to the appropriate areas and repair the brain. Thus far, I can't tell you that I'm optimistic that anybody has other than for very small areas of stroke, anything that's likely to be on the immediate horizon. On the longer term, it will most certainly become feasible to at least patch areas of the brain that are damaged. If there's a massive stroke, we're not even close to being wise enough to think about rewiring a whole brain. For small areas of infarction, absolutely.
One of you.
Well, first of all, it was very, very interesting, so thank you. Do you have any timeline on human trials, especially concerning the cartilage regeneration?
Which one? Cartilage.
Cartilage regeneration.
Well, yes, we are. So what we are doing, it's a very costly process, because in order to translate the cartilage regeneration technology, the FDA requires that we test it in large animals. For example, you are required to do studies in goats, in sheep, and so these are very costly. So what we have done is we have partnered with a company, a large company that is interested in translating, and according to their plan, they would like to think about maybe 2 or 3 years to get to phase 1 trials.
With that.
So I had a question for Dr. Stupp. You mentioned that the nanofibers may be biodegradable. What, in fact, you mentioned that they are. How did you assess that?
Well, it's possible to, we know it from both in vivo experiments and also in vitro experiments. So in vivo, for example, in the spinal cord injury models that Dr. Kessler worked on, we were able to see directly how long the fibers survive, and we were able to observe that after about 6 to 8 weeks, the material was effectively gone, right? 4 to 6 weeks. In other models that we have done in different types of animals, different tissues, we find that some of the fibers survive about 3 weeks. We have a set of fibers that we're studying right now in subcutaneous models, and they last exactly 3 weeks. And then, of course, you can do in vitro assays also to determine that the fibers are easily degraded by proteases, which are the enzymes that break peptide bonds.
The way you look at it is you hook up a molecule to it that's called fluorescence. It means it will glow under the microscope. Then you can actually just follow the fibers and see how stable they are. So you can follow the disappearance of the fluorescence and actually see the fibers degrading.
This may not be relevant to what you're talking about today, but I read today that the NIH has approved, we'll finish my question.
Yes, it's very relevant. Under the former president, who really had no concept about how to deal with the issues regarding stem cell biology, he had a very illogical and unfortunate position that was changed by President Obama, who said that the NIH would now allow research dollars to be used for embryonic stem cell lines that were created under conditions that were considered ethical and moral conditions. So today it was announced that the NIH is now approving a series of new lines that have met all those criteria, which expands to researchers the number of human embryonic stem cell lines that are available for us to use. I happen to already in my stem cell facility have some of those lines. I set my facility up with what I call the kosher half and the non-kosher half. So, you know, in the kosher half, I was only using the NIH-approved ones, and now I can use those lines in both sides of my lab. There are still a number of restrictions in our ability to create new lines and for doing some things called nuclear transfer. So the policy hasn't been completely changed, but it's a very, very good step in the right direction. It has a big influence on our stem cell lab.
Other questions? So I've saved a couple. Oh, go ahead.
What about the chance of overgrowth and overgeneration of you know, what you were looking at?
It's a wonderful question. One of the problems that, for example, in spinal cord regeneration, if we get fibers to regrow, that's not enough. You have to get them to not only grow, but to hook up in the appropriate way. Now it happens, nature has already tells us that if you can get them to what's called the target, they'll mostly hook up the right way. Most of you have seen people who've had something called the Bell's palsy, where one side of their face becomes weak. Most of the time the fibers grow back the right way. Sometimes they grow back incorrectly, so you get something called the synkinesis. What does that mean? That means if somebody smiles, their eye will blink. Or they get what are called crocodile tears, which means they'll smile and they'll get tears, because the fibers that are supposed to go one place grow back the other place. Our big fear in the spinal cord is that when we get fibers to grow, they're going to hook up in the incorrect way and give people pain. Because if you start getting the wrong kinds of signals going back to the brain is going to interpret that as pain. So we've looked pretty carefully thus far in our animals for any evidence that we're creating a painful kind of syndrome. We haven't seen it, but it is something that is that we're actually quite fearful of.
Yeah, if I could add to that question, in a more general context, in regenerative targets, the possibility of overgrowing is of course ameliorated by the fact that the fibers are degraded. So they're sort of self-limiting, if you will.
I didn't even think of the pain concept. I was more thinking of maybe wrongly cancer or tumor growth, so overgeneration into undifferentiated cells that could potentially be cancerous.
Well, I think Professor Stupp made the appropriate point. That's the whole point of having something that's biodegradable, so that it only signals for a period of time and then it stops. And the body has a lot of systems in place actually to control growth of cells that aren't mutated. So that's probably not as big a concern.
I'm just very curious when you're talking about things that are so small as you are, how you physically create these and manipulate those in a lab when you work with something so small. You showed the image at the electron microscope, so that's how you view it once you've created it.
That's right. So we use microscopes to view them, to image them. And what we do in the lab is we make molecules, but we make those molecules using the same techniques of organic chemistry that have been used for more than a century now. Going back to German chemistry in the late 19th century. So we use this, we basically use synthetic chemistry to make the molecules, but then the design is planned so that they will self-assemble into these fibers on their own. So what you do next is you image them, you use powerful microscopes to see them, to verify that they have assembled in the way you've designed the system to assemble. But also we have other microscopes that allow us to probe them. There are many of the variety called scanning microscopes, where you can actually measure the properties of single fibers. So you could actually go in with a probe and touch one fiber only and measure how stiff it is or how soft it is. So its instrumentation is, in fact, the genesis of nanotechnology has a lot to do with the creation of new instruments that were the result of the microtechnology revolution. So microtechnology led to new scientific instruments, and those new scientific instruments led to nanotechnology.
So you create these on a lab bench?
Oh yes, on a lab bench.
Like anywhere.
Yeah, if you go to my lab, in the medical school or in Evanston, yeah, you'll see benches and hoods and rotary evaporators and Schlenk lines and all the things that you expect to see in a chemistry lab. And then lots of fancy instruments to characterize what you make. And then, of course, microscopes in central facilities.
If that is actually an offer, then I would accept it.
Okay.
Other questions? So I'm going to take the moderator's prerogative. And there was the cancer question. So the question I would have is, can this technology be used on cancer that already exists, not one that you create, and try to regulate that?
So the answer is we're already doing that.
Okay.
So there's the concept of what's called the cancer stem cell. Why do cancers recur? The surgeon goes in, takes the whole thing out, I got it all, and of course, then they recur. And the concept of medicine now is that most cancers arise not from the differentiator, the regular cells in the tissue, but those stem cells that are sitting there. These cells already have the characteristic of being immortal, able to divide indefinitely, so it takes less change in those cells to convert them into a cancer cell. So there's what's called a glioblastoma is the worst kind of brain tumor you can get. It's the kind of tumor that killed Kennedy recently, for example. So we have taken 20 patients at Northwestern Memorial Hospital, established brain tumor cancer stem cells. And we're actually using some of these very same materials right now to differentiate those cells. The goal being, if you can take this stem cell, the cell that makes the tumor come back, and force this cell to stop dividing and differentiate and become a more normal kind of cell, then basically you have a way of preventing the recurrence of cancer. So it's something that we're actively doing with this specific technology, and of course other kinds of signals as well. So it's a very good question.
One more.
Is there one? No more questions? Thank you for saying my question was very good. I was going to end this by saying that I've listened to Sam and Jack talk for many years, and this set of questions was an impressive set of questions. So I would like to say thank you to the audience, and once again we should thank our speakers.